we're going to go through one example here of synergy for microbes. Microbes can form biofilms, and biofilms are like small communities or little environments for bacteria that attach to the surface. And so they'll form like a little slime layer and a group of microbes all together to form a multi-layered conglomerate of cells and kind of a intracellular material, okay? And by forming this, they can all work together. They're still their own entity. They don't have to be together, but when they're all working together, they're much more efficient. Okay, so we're going to go through this little flow chart here. We first start with a pioneer bacteria. A pioneer is the initial bacterium that attaches to the surface. So maybe you have like, I don't know, a catheter or a surface in the hospital, something like that, okay? And that initial bacteria shows up on the surface. The pioneer will then secrete proteins, extracellular materials to kind of protect them from drying out, help them stick to the surface. Maybe they're going to make a slime layer or a capsule or something like that. It's all excreted. And while they're doing that, they're also propagating. So they're dividing, and now there's 2 cells, now there's 4 cells, and so on. This allows, with the slime layer in green and the cells inside of it, for them to release chemicals, these little red dots that they can use kind of to signal to each other. Quorum sensing is the ability of bacteria to regulate gene expression in response to bacterial density. So the cells can go in here and they're in their extracellular material, and there's a bunch of them. They can send messages to each other, little red dots, through quorum sensing to go, hey, cool, we have a ton of food, let's divide. So they can all start dividing. And then maybe the food's running out, they can go, oh no, maybe you guys need to kind of branch out and be a pioneer and leave now, or slow down the metabolism and things like that. So they're not a multicellular organism, but they are working together as a community. In many biofilms, other species will join. So you can get, you'll see in the picture here, we not only now have the little pink bacillus, but we have purple bacillus, and we have a blue coccus showing up, and they can still sense each other, and they'll continue to secrete extracellular products. This will serve as a constant source of bacteria. So we'll have one area that has a biofilm with lots of kind of safe bacteria, and some bacteria will be leaving and further propagating onto other surfaces. Okay, so the last section of the chapter is on how do we study bacterial growth in the lab. Okay, so before we get to that, it's important to talk about how bacteria divide. Bacteria divide through binary fission. So they'll start with, you have a single young cell, and it's maybe getting quorum sensing signals that says, hey, there's lots of space, we better divide. So that cell will replicate its chromosome in pink, and it will replicate anything else it needs to, separate the DNA to either ends. It'll make a protein band in the middle, a septum or kind of a splitting spot will start, and then we'll have 2 bacteria that are identical from the one we started with. Okay, so one big takeaway from this is when bacteria divide, we're going from 1 to 2. And if we went again, we'd go, this one goes from 1 to 2, this one goes from 1 to 2. We'd be going from 1 to 2 to 4. Okay, so we can predict and count. It's not random. We're counting very methodically how many bacteria are there. We can also determine experimentally, and probably in the literature for many microbes, the generation or the doubling time. The doubling time for a microbe is the time it takes from one fission cycle to occur. So going from 1 cell to 2 cells. So this would be A generation time would be if this took 16 hours for this step, or from here to here took 2 days. That would be the generation time. The average generation time for bacteria is 30 to 60 minutes. Now, some are faster, so they can do it every 10 minutes, get with it. And then there's some that are much slower on the scale of 10 to 30 days. That would include species like Mycobacterium tuberculosis that causes tuberculosis, part of why some infections are really slow to show up and hard to track when you are initially exposed. Okay, so as I was mentioning, since we know binary fission, it's very predictable going from 1 cell to 2 cell to 4 cell, we can mathematically work backwards to figure out how many bacteria are in a broth, how many were in the original sample, and so on. Okay, so we can kind of follow the pyramid here and go, okay, we have 1 cell, now we have 2, now we have 4, now we have 8, 16, and so on. Okay, very predictable. You could see how we could get exponential growth. Okay, so it kind of chugs along at a slow rate for a while, and then pretty quickly it can get away from you. Bacteria, though, don't maintain that perfect growth rate forever. They can't double indefinitely in a closed system because eventually they'll end up running out of their essential nutrients that we talked about last time. Typically, bacterial growth follows this standard growth curve. And this is a graphical representation of how you would expect a population to change over time, at least in the lab. There are 4 main phases that it will go through. The lag phase, where there's very few cells, but it is dividing and there's enough nutrients. There'll be an exponential growth phase where that growth gets away from them and we have a bunch of cells and we can see visibly in the tube evidence of growth, whether it's turbid or cloudy or there's clumps on top. You can see that there have been bacteria dividing. Then you'll enter a stationary phase where they're starting to exhaust their nutrients. You might get some die off in the bottom of the tube, but they're still dividing. And then the death phase, where there's some bacteria that are remaining viable, but most of them are not going to make it because there's not enough nutrients. So that was if we were to grow them in a broth, but what about on a plate? You can do a standard plate counting method to actually count the number of bacteria that were in your original sample. To do this, you add a tiny number of cells into your sterile broth. So you would take a small sample, maybe you're looking at if there are fecal coliforms in water, or you want to know if your food is contaminated. You take a small amount, add it to a sterile broth to grow it up. You'd incubate it in culture for several hours, so it would follow the growth curve we saw in the last slide. And you could sample the broth at regular intervals. With each sample you pulled off, you could put it onto a plate of agar, let it grow for a defined amount of time, maybe 24, 48 hours, count the number of colonies or dots of bacteria on the plate, and then you could work backwards to figure out how fast they were growing, how many of them were there to start with. A key term that comes up when we count bacteria on a plate is CFUs. These are colony forming units. So you assume that each colony that is visible on the plate was 1 bacteria initially to start with. Okay, so to see how this would work, you would start with your sample here and add it to your sterile broth. And then you could grow the broth up over time, and you can see as time passes, it becomes more turbid or cloudy, indicating you have more bacteria. But again, I can't count it by eye. I can't look at this and go, oh, there's 2,300 bacteria in this tube. To do that, I have to use the plate. So you can add a little bit to the plate as you go along, and we can count the colonies to determine how many there were in the original sample. Okay, so there are a couple other methods that you can use to analyze the population size. But before we get there, we need some comedic relief. So here's a comic for you. You have two scientists, and one says, "I've discovered a talking amoeba." So he looks through the microscope, and it's 2 + 2, 5 - 4, 9 divided by 3. Bam! Why'd you do that? Well, I just wanted to get to it before it multiplied. So we can analyze populations and see how quickly they're multiplying, how many are there, and kind of extrapolate out how many will eventually be there. One method we can use for this that's really quick and easy is to analyze turbidity, or cloudiness of the nutrient source. So if it had low turbidity, a pure sterile broth, I would expect to look very clear like this. You can see through it. And if we put it in a spectrophotometer and shine a light through it and get some readings out of it, it's almost all the light gets through. It's all transmitted, low turbidity. That would tell us that there are not very many bacteria in that broth. But maybe you added some bacteria, grew this up overnight, came back and it looked much more cloudy. Then if we added it into the spectrophotometer, the light wouldn't be transmitted as well. It would be much lower, which would tell us, oh, we have more bacterial growth in the sample. Again, this is much more difficult than plate counting to get the actual number of bacteria. But you can get a relative growth of, okay, hey, we had nothing, now we have a little bit more, oh, now we have a lot. So it's kind of relative amounts, but not exact numbers. You can also do a direct cell count. This uses a special slide, special microscope slide that has a grid on it. And you can add a couple of drops of bacterial solution to it, very defined amount of like 10 microliters, 15 microliters or something like that to the slide. You can count all of them up, and then you can figure out, okay, I had 10 bacteria and 15 microliters. Therefore, if I had 1 milliliter, you can kind of work backwards and figure out how many you have in your overall sample. The one downside of this, though, is that this does not account for the number of live cells or dead cells. So when you look at these, if a cell is living and multiplying, you can't tell versus that one and one that was alive and no longer is. So it's much harder if you're trying to get viability. It's just going to tell you how many are in the broth as a whole. You can also use a flow cytometer. So this can count the number of bacteria. It will be pulled through a machine and it will count them as they go by. And it will tell you the number of cells. It can tell you how big the cells are. So if they're all uniform, or maybe they've been infected with the virus and now they're suddenly bigger and have multiple nuclei. It could tell you that. It could tell you if they're alive or dead. So it would tell you how viable they are. You can also label them, like in the picture here, with different antibodies. And you can sort them and go, okay, I want all of them that have this cell surface marker over here, and the ones that have this other receptor over here. The final method you can use is a Coulter counter. This is a device that scans the fluid. So if you have your broth with bacteria up here, you could add it to the counter, it would go through the fine tube, and as each of the bacteria goes by, it counts it with this electronic detector, and you can count all the bacteria in a sample.